Hi everyone. How are you guys doing? I hope you are doing great. And how was the exam and how was your weekend? Hope it's fine. So today's lecture is on microbial nutrition and growth. So these are the objectives. First, we will learn to list the 6 essential macronutrients and state the overall use for these macronutrients in the microorganism cells. And then we will also name one of the essential micronutrients of a cell and state the overall use for micronutrients in the cell. We will also describe briefly the process of binary fusion and define generation and doubling time and explain how it leads to exponential growth in bacteria. We will compare and contrast the 4 phases of growth in bacteria growth curve and then draw a graph representing these phases of the growth curve. We will then describe how we commonly quantify bacteria use using key terms in both ways, basically 2 ways, and explain the key terms used to express a microbe's range of growth, temperatures, and oxygen requirements. Lastly, we will summarize each of the environmental factors impacting growth and its applications, and name the type of extremophiles that live under these conditions. So first, microbes, like any other humans, require food to eat. So I'm talking of all living organisms, except that of viruses and prions, which do not qualify as cells. So, what do microbes eat and how do they grow? So, like any other living organism, they will require nutrients from the environment and they use this for their cellular activities. So, they require different kinds of nutrition or nutrient. One being essential nutrient, that is any substance which is in elemental or molecular form and then must be provided to the organism. And then we have macronutrients, which is required in larger quantities. These are nutrients which are required in larger quantities and they normally play larger roles in these bacteria during metabolism and the formation of cell structure. And we have micronutrients, which are mostly needed in smaller amounts and they are used in enzymatic reactions and functions in the microorganism, basically helping to maintain protein structure. And we have organic nutrients, which contains either carbon or hydrogen atoms, usually product of living. They are usually product of living things, and the bacteria use this basically for the formation of proteins. And then we have inorganic nutrients, which are basically atoms and simple molecules that contain atoms other than carbon and hydrogen. So there are 6 essential macronutrients, which is basically carbon, nitrogen, oxygen, hydrogen, phosphorus, and sulfur. So carbon is used, basically helps in the structure of proteins and nucleic acid and lipids. Nitrogen is also required for the structure of proteins, nucleic acid, and ATP. Oxygen is required for the structure of proteins, lipids, carbohydrates, and nucleic acid. Hydrogen is also required for hydrogen bonds between amino acid, lipids, nucleic acid, sugars, and also help in maintaining pH and respiration. And then phosphorus is needed for the structure of nucleic acid, lipids, and ATP, and is mostly found in coenzymes. And then sulfur is needed for is part of the component which is needed for vitamins. Some of the vitamins, including vitamin B1 and some amino acid, such as methionine. So, what are essential micronutrients? We already described micronutrients are needed in smaller forms, and they are usually needed for enzymatic function and maintaining of cell signaling and also protein structure. So, some of the essential nutrients include iron, zinc, manganese, potassium, magnesium, and calcium. About 30% of this is required for proteins, serving as cofactors. So let's talk about the nutrient acquisition of these bacteria, of these microbes. How do these microbes obtain their nutrients? So their nutrient acquisition typically depends on the microbial type and where they grow. For example, if you take fungi, you can have saprobes or parasites. And depending on how depending on what they are. For example, parasites will require their nutrients from their host compared to saprobes who will be requiring their nutrients from dead organisms. A second example will be bacteria. Whether it's found in the soil, it will require their nutrients in the soil. If it's been found in marine environment, it will require their nutrients from the environment. And if it's found in humans or their host, they also require nutrients from their host. So, as they get this nutrient, they have to eat. Once they are eating, then they have to grow, same as every living organism. So, how do bacteria then grow? Or how do the microbes grow? So now let's first tackle bacteria. So in bacteria, there is a self-division. They just grow binary. So there's a division. Basically, the bacteria just divides or reproduce two of itself. So now let's look at here. Step one, we have a young bacteria here, a young cell here, just young. It's eating, everything seems super fine. And then it starts growing. Basically, what it does is duplicate the nucleic acid in any other organelle in here, and the old chromosome and everything moves to the other side, and the newer one is here. Once the whole duplication is completed, there is a formation of protein bands in the center of the cell that separate both the old and the newly formed chromosomes. And then septum formation begins where the cell actually starts to divide, producing two different cells. And eventually, once the septum is complete, the whole bacteria then separation is super completed, and now you have two different two baby bacteria or two sister bacteria, which are basically coming from the same one. And then during binary fusion, as we've discussed earlier on, there are numerous enzymes or numerous macromolecules and micromolecules or micronutrients which are being involved. For example, if you take some of the micromolecules which are essential for the functioning of enzymes, here you can see that a lot of these enzymes present here are using macromolecules which are coming from ATP to help synthesize proteins for basically the cell wall, and also other proteins which are needed for the cell wall during division or during the whole formation of septums in this bacterial binary fusion. So you can really appreciate the importance of nutrients when it comes to growth or binary fusion in bacteria. So let's talk about the rate of population growth. So generally, the generation time or the double time depends on the number of fusion which is being completed, and each cycle doubles basically the population. So if you can see here, one bacteria divides into two cells, and then the next cycle the two cells also divide into two separate cells. This one also divides into two separate cells, and they keep dividing over and over, just basically in the exponential of twos. So typically bacteria population growth happens around 20 to 60 minutes, and it has been shown that like the shorter is 10 minutes, the longest comes in for days, and it's being influenced by incubation time. So exponential growth is basically the increase in of bacterial population where the number of cells doubles at a constant rate per each generation. So as long as these conditions are favorable, the bacteria keep doubling and they keep growing. And to basically understand or study this, there is a term we call the growth curve, basically a predicting pattern of growth in a population, typically measuring the growth in a broth over a broth culture over some time. So to now understand the growth curve or the way in which we can predict the growth of the bacteria, we present you with this population growth curve. First, we have the first stage, which is the lag phase. This is basically a flat line. This is basically the flat line at the end of the graph. This is basically when the newly inoculated cells are there. They are now getting used to the media or the environment, and basically they need some time to adjust and to synthesize any enzyme that is needed for duplication. And then we move to the exponential growth phase. This is where the bacteria have now finished relaxing. They are used to the environment. They are now synthesizing a lot of enzymes, and they are making duplication or doubles of themselves. And there's available nutrients, so they grow super fast. And then they move to the stationary stage where the bacteria has finished growing. There's a lot of them, and they entered into a survival mode due to limiting factors like nutrients and space, and the cells stopped growing rapidly as they were growing in the exponential phase. And then we move to the death phase, where due to the limiting factors intensifying, the cells begin to die at a high exponential rate. So now that we understand how the bacteria grows and the nutrients that are being required, we also need to find a way um, to measure the amount of bacteria present at each phase. To do that, we first have a first method, which is the turbidity. The turbidometry or OD. Basically, the basic mechanism is that here is just an empty media. When you shine light through the media and you kind of measure the intensity of the light through it using this um, machine, you should be able to, the whole light should be able to pass through the liquid because it's clear. Once the liquid is turbid, light is not able to go through, and then you can now justify that there's a lot of bacteria in here that is growing and is turbid. For that reason, the light can't go through, and then you can quantify that as a high bacterial growth. This turbidometer measures the turbidity or the cloudness using 600 nanometer wavelength passing through the sample, as I've explained earlier on. Secondly, you can also measure this by counting the bacterial colonies. And that's what we've talked about so far in class in lab. You first have to perform count the total number of cells by doing a serial dilutions where you have a concentrated growth bacteria here, you perform a dilution. So you pipette, let's say 100 microliters here, you mix it up, move another 100 microliters, mix it up till you get to the last stage, and then you plate equal quantities on the bacteria plate, and then you later come back and count them using a colony counter, or you count them manually and calculate how much colonies are being formed. That is the second method. So we can practice this, and we can also discuss this also in class. So you should practice this on your own. So now that we understand how bacteria grow, we can also quantify how many of them are being present. We also have to understand how environmental factors impact the growth of bacteria. So several factors affect the growth of bacteria. These include temperature, gases, pH, osmotic pressure, hydrostatic pressure, radiation, fluid flow, and also other microbes. And then we also have chemicals, which are basically the nutrient availability, which we've already discussed, and also antimicrobial agent, basically agent that kills the bacteria itself. So now let's first talk about temperature. So temperature, we have a huge range of temperatures which are needed for the growth of each microbial species, depending on the type of microbes we have. So these there are small chemical differences in bacterial membrane which affect their fluidity, allowing them to thrive at different temperatures. So temperature will impact them if and how fast the bacteria are supposed to grow. So let's say a bacteria can only survive in high temperatures. You grow them in lower temperature, they can grow super faster. And then the minimum temperature, so basically you have a minimum temperature or the lowest temperature which they can survive. We have the maximum temperature, that is the maximum temperature they can survive, and then they have their own ideal temperature in which they can grow. So these different temperatures are being termed as the cardinal temperature. So cardinal temperature, I'm showing one here in a graph. So cardinal temperature encompasses the maximum, minimum, and the ideal temperature in which the bacteria can grow. So, for example, if you take cycrophilic, which are bacteria, which microbes, which require lower temperatures in order to survive. For example, here you can realize that when you keep these cycrophilic in a normal refrigerator, most of them are able to survive. When you keep going lower of a temperature or lower freezers, most of them start dying. And then we have the mesophilic, which requires normal room temperature to survive. And you realize that at normal room temperature or body temperature, and normal room temperature and also the body temperature, you realize that most of them are surviving. And as you move to the colder side, they die, or you move to the hotter side, they also die. And then you have thermophilic, which are bacteria which require higher temperature to survive. And you realize that the same thing also happen. You come to the colder or the minimum place, they die. And when you go to the maximum also, they also die. So now let's talk about mesophiles. So majority of them medically are the majority of them are medically significant organism, and they have a temperature they require a temperature ranging from 10 degrees to 50 degrees with an optimum temperature around 20 to 40 degrees. Most of them are human pathogens, and you do realize that from 30 to 40 degrees is basically most of where the human body can survive with this temperature. And then we move to extremophiles. This basically consists of the psychrophiles and also the thermophiles. So the psychrophiles, they love cold temperatures, and then they can survive from minus 15 to 15 degrees and cannot grow past 20 degrees. If you do realize that if you have a microbe which is a psychrophile and you think freezing your food is gonna kill them, you realize that it definitely wouldn't work, as they will still survive in refrigerators and will still survive in your food. And then we have the thermophiles, which really love hot temperatures, and they are mostly found within springs and hot springs. They require a temperature of 45 to 80 degrees. And then we have the hyperthermophiles, which require 80 to 113 and cannot grow in optimal like 50 less than 50 degrees. And we have these microbes, which are more present in volcanic water, soil, and submarine vents. So now that we know the temperature these microbes require, what about the other category, which is gases? So when in relation to gases, some microbes require oxygen and some do not. So they need this oxygen to perform cellular reaction that will basically produces several toxic oxygen products. This will include superoxide, hydrogen peroxide, hydroxyl radicals, and a lot of other things present. These microbes, which require oxygen, falls into one of these three categories. Categories. One is first category is that these microbes that fall under the first categories will be able to use and can detoxify oxygen. The second microbe can neither use oxygen nor detoxify it. The third microbe do not use oxygen but can detoxify it. So now let's talk about the groups of microorganisms based on oxygen requirement. The first one is aerobic. Basically, they use oxygen and they use oxygen and they have enzyme that process toxic oxygen products. So we have obligate aerobes. Obligate aerobe, basically they cannot grow without oxygen, and many of them are fungi and protozoans. So as you can see here, in you have an obligate aerobe bacteria. They if you put them in a media like this, they will always grow closer to where the oxygen source is. On the opposite side, when you have facultative aerobes, basically they can use but do not require oxygen, and they are capable of growth in the absence of oxygen. Many of them are the bacteria, some of the bacteria that we just cautioned in our past lab experiment. And but comparing that with the obligate, which require oxygen, so they grow closer to the source of oxygen here. The facultative, they can survive with or without oxygen. For that reason, they just as you can see here, they just grow either closer or far away from the oxygen. The third group is anaerobic. Basically, they do not require oxygen, and then they are strictly anaerobic and they die in the presence of oxygen. They often live in deep mud, lakes, oceans, or soil, and they include bacteria, archaea, protozoa. And then we have the other fourth one, which is micro aerophile. They do not grow at atmospheric concentration of oxygen. They just require little amount of oxygen, and some of them are bacteria and also protozoa. So as you can see from here, in this instance, the obligate microbes which do not require obligate anaerobe which do not require oxygen, you can see that they are growing far away from the oxygen source in these two. And then the micro anaerobe, which requires just a little amount of oxygen, they are not super close to the source of oxygen or far away from it. They are just minimally close to that. The fifth one, which is aerotolerant anaerobes. Basically, they do not use oxygen. They can survive and grow to limited extent in its presence. They possess alternative mechanism for breaking down toxic products, and they are mainly bacteria and also some archaea. And as you can see from here, they sorry. And as you can see from here, they are growing evenly spread throughout the whole media. So now that we've discussed microbes that require oxygen, what about carbon dioxide? So with carbon dioxide, we have capnophiles. These are microbes that grow best at higher carbon dioxide concentration, and mostly you find them within 3% to 10% of carbon dioxide concentration. They include the oral and gut bacteria. Then they have some, we also have some which are respiratory tract bacteria. Some include archaea and fungi. And as you can see from here, this is just a clear example. And we also have this also in our lab where we grew some bacteria in the presence of carbon dioxide. We also have bacteria that requires a certain pH in order to grow. And most bacteria prefer a neutral pH. However, we have alkalinophiles, which prefer basic or alkaline environment. And most of them, you can find them in hot pools and soils with high level of basic minerals. And most of them are archaea. And also, we also have some fungi and protozoa, which are mostly rare. And we have bacteria that decompose urine in UTIs, which basically creates alkaline conditions. Opposite to that, we also have acidophiles, which are bacteria that require acidic environment in order to grow. Many of which are mold and yeast, and they tolerate acid and they tolerate acidic environment. Some example is what we've seen from here, which is thermoplasma, which is a group of algae who survive in hot, cold piles and can reach a pH of 1 to 2, as you can see from here. Then we have microbes, which can be differentiated based on their osmotic their requirement for osmotic pressure. So most microbes prefer or require isotonic environment, but we have osmophiles, which require high solute concentration and have high cell membrane modification to survive these. So under these, we have facultative halophiles, which are basically resistant to salt. These are Staphylococcus aureus, and as you can see a picture showing the Dead Sea where it's highly concentrated with salt, and you can clearly find microbes over there. We have obligate halophiles, which require high concentration of salt for them to grow, basically 9 to 25% of sodium chloride. And we have extreme halophiles, which require salt to grow, and they will grow in about 36% sodium chloride, such as in inland sea, salt mines, and also in salted fish. For example, this is a good thing for you to note that the Dead Sea have over 1,000 to 10,000 archaea per ml, and basically these are about 33.7% salt concentration compared to that of the ocean, which is 3.5%. Then we have microbes, which require certain which require hydrostatic pressure to survive. And then hydrostatic pressure basically is the pressure exerted by water or fluid at equilibrium at point in time due to the force of gravity. So most microbes prefer atmospheric pressure. So we have a certain group of microbes called barophiles, which strictly are adapted to up to 1,000 times deep sea atmospheric pressure, and they are mainly archaea and bacteria. and some other types too. And basically, in this picture, you can see that even if you watch those these deep sea creatures and anime and all these things they show on TV, you realize that they have microbes deep down under the sea. And also something you have to take note is that because these microbes which require this high atmospheric pressure, they are used to living down deep sea. Once you bring them up, they will rupture or just get basically they will get ruptured when you bring them up because of the change in atmospheric pressure. When you take them out of the sea bottom, they are gonna rupture. Now the sixth one, which is radiation. So we get radiation from sunlight, basically UV. This group of microbes which require radiation, basically, they have photosynthetic ability. Basically, they have photosynthesizers that they can use to visually they can use visible light rays as energy source. And these include algae and cyanobacteria, which I'm showing in this picture here. And they also We also have within this same category, we also have some microbes which do not have the photosynthesizers, and these microbes are actually degraded or damaged when you expose them to light. And some radiations are used as antimicrobial treatment in the chemical field. You use them towards the microbes which do not have the which do not have the photosynthesizers. But if you use radiation on the photosynthesizers, basically you are just giving them food and it will never work against them. You can only use UV or radiation against the non-photosynthesizers because they don't have it and they will die. So we also have under number 7 is fluid flow. So many microbes live in fluid environment. And then this fluid flow basically impact their population in attachment, nutrient access, and motility. And as you can realize that some microbes live in flowing water. Once the the liquid is flowing, they basically need to be flowing all the time. And some microbes need to attach also to a surface in order to grow. And you can clearly appreciate if you put a microbe that needs to attach to a surface to something that flows, whether it's blood or it's a liquid or just a pond or something, that microbe wouldn't survive. The same way, if you take the ones who require flowing to in order to survive and put them in a static environment, they will also die. So we also have microbes that have synergic or antagonist relationship between other microbes and that can influence their growth. So now let's talk about synergism. So basically working together, as we discussed earlier, some microbes will form biofilms where they will attract other microbes to come and join the community and they provide nutrients for each other. And then we have the antagonism, basically where we have microbes secreting toxins in order to kill other microbes when they are basically competing for nutrient or survival. And one clear example is here. You have a group of microbes growing together. We have to, so here we have Staphylococcus growing, and we have penicillin, which is being secreted by these molds. And then the penicillin is basically killing all this Staphylococcus that is being present here. So as you can see, this is the zone of inhibited growth, as penicillin is more higher here, and then Staphylococcus surrounding this whole area is dead. So now let's practice. Try and find the microbial nutritional case and practice on model and answer these. Please remember to come to class tomorrow as we have tomorrow's lab, and try and do post lab and after lab. Try and do everything on time. Because there won't be much time for you to do. I make sure I extend everything 1 week for everybody to get it done. I'm not gonna extend it past another week, so get everything done on time. See you tomorrow. Bye.